I'm happy to be here today to be the token not snake guy. And very uncreatively, as Mike has already said, I'm talking about social parasite ants and how they're investing in venom. I'm happy to be talking to a group of venom biologists and to go after Navanil, who already gave an excellent explanation of what venom is, because now I don't have to spend several minutes explaining why venom is super cool. So instead, I just want to orient us to where ants are in the tree of life. This is the phylogeny of all the venomous animal taxa. You may have seen this before in Schindel et al. 2019. And we're interested in the Apocrita, which is right here. Apocrita is a clade within Hymenoptera, the sawflies, wasps, bees, and ants. And if you were to expand that out and see what taxa actually live within Apocrita, you get something that looks like this. A lot of the narrow-waisted wasps. Down here at the bottom, you see the bees and the ants. And as the title of my talk might indicate, I'm interested in the ants. There are about 16,000 species of ants. That's a lot of ants, at least the last time I counted. And their venoms, I think, as with all venoms, are pretty neat. So almost all ant species are venomous, but their venom comes in a lot of different forms. Some of them actually shoot this formic acid. I think the acid is named after the clade from the city, not the other way around. And they'll do this, they'll point their butts up in the air and just fling acid out at any would-be predators. Some of them, like the army ants, have a proteinaceous venom that they use more like a trail pheromone rather than as a way to kill or subdue or defend. Army ants instead just rip things apart with their mandibles. And some of them, such as this fire ant, have secondary metabolites called alkaloids. And that these secondary metabolites are what I'm interested in because my study system has venoms composed of these alkaloids. So let's talk about alkaloids. Ant alkaloids are sort of like chemical multi-tools. Ants use them in kind of every way I feel like you could conceive of uh, bio piece of biochemistry. So an antimicrobial sort of medicine as offense to kill prey, as defense to deter predators. If anyone's ever been stung by a fire ant, you know how effective their venom is at deterring predators. And as a form of communication. So when they produce this venom, where they deliver that venom, their sisters can smell that they have and will then rush over to deal with whatever problem is arising. Ants use these in lots of different ways. So as an insecticide to kill prey, as an antimicrobial. And I think the funniest way they use this when they're communicating is they will kind of stand up, they push their legs up and then stick their gaster, their butt up in the air, and then they produce little droplets of venom they fling around as a way to repel other ants that would be competing with them. So they use it to dominate resources as well. And we see evidence for this in all of the different venomous genera or venom alkaloid containing genera, but I'm interested in Megalomyrmex, the big ants, some of which are not so big. There are about 45 species of Megalomyrmex, if I'm remembering correctly, at least described. There are a lot more lineages we haven't described yet. They range from the southern tip of Mexico all the way through Central America, down into South America, kind of down into the southern parts of Brazil, potentially further down. I'm not sure if anyone's really spent a lot of time investigating beyond Brazil. And these ants, other than their venoms, a lot of people think they're really cool because while most of them are the free living, kind of walking around doing ant things like M. leoninus up here, about a quarter of the taxa are these social parasites. And instead of walking around and being normal ants, instead what they do is they find a host fungus growing ant and they parasitize them. So they'll take garden, they'll take host brood, and just live off of that. So within Megalomyrmex, they're kind of broadly split up by morphology, and Within there, the social parasites are a monophyletic clade in the Sylvestrii clade on the bottom right. Social parasitism manifests in three different strategies. We have the guest ants, the ones that live within the host colony. We have the thief ants, ones that live nearby the host ant colony, but not within, and the sort of raider or Viking ants, which will adopt a mix of strategies depending on how the host decides to react. So they might live outside of the nest, they might live in a chamber that connects to the nest, but not directly, or they might even live sort of within the nest along the periphery. These strategies also vary depending on how obligate or facultative they are. So some of the parasites don't need a host. If there's a host nearby, they'll use one, but otherwise they'll just behave like a normal ant does and forage. Some of them are obligate, so they need to have a host, otherwise they just die. Despite all of these differences, megalomyrmex are really united in delivering their alkaloid venoms. They do this. Ooh, sorry. These are supposed to be videos. So they'll gaster flag, or the twerk of doom, as I like to unprofessionally call it. They fling out that venom everywhere. They will sting when they're dealing with things that they can't get their stinger into. Of course, they still topically apply their venom. They get very mad when they do this. It's the F around and find out. And the guest ants even like to drop venom down onto their host's garden, or what your drunk friend did that one time you let them crash on your couch and they decided to just not leave. So all really cool behaviors. And I was really interested in understanding kind of how this venom changes across the evolutionary landscape, and if there are really any differences within how the parasites versus the free-living ants invest in their venom. And I was kind of thinking of it as the free-living use their venom a lot to deter competition, sometimes to kill prey, but they spend a lot of time just foraging things like seeds and nuts and fruits. And so for them, their venom really equals their dinner, where they don't need to have their venom because if they can find a resource early enough, they can still get extract a large amount of it. But for the parasites, especially for the obligate parasites, use of their venom is their life. They need to use their venom in order to infiltrate and integrate within their colony. And if their venom isn't effective, well, they just die. Here we have an example of a parasite queen inducing host queen submission. So the parasite queen, if she's bothered, they try to just stay away and leave the hosts alone. But if she's bothered, will sting a host, and then the hosts tend to curl up because it turns out getting hit with venom is maybe not the most fun thing to happen. And I really wanted to investigate this investment in venom in three ways. So first, understanding exactly how the venoms are composed and if there are any differences there. Investigating potential ecological function, because while I've outlined a lot of different ways they use their venom, nobody has really spent a lot of time in particular with megalomyrmex and the social parasites. And thinking about the larger pieces of their venom phenotype, really their physiology, how much venom they're actually holding at any one time, because how much venom they hold informs how much venom they could deliver. We're going to go with those in order. So first things first, the venom alkaloid composition. This is a 5-gene phylogeny. I think it's 2 mitochondrial genes, 3 genomic genes, built here using as many different megalomyrmex as we could find. And then we also dumped the ants in methanol and analyzed their venom alkaloids using gas chromatography mass spectrometry. We see that there are 5 different classes of alkaloids. Some people argue that the pyrroline and pyrrolidines should be paired together since the difference is a double bond with the nitrogen. And that's a pretty fair comparison, I think. And while these classes are found in other ant genera, no other ant genus has all 5 of these classes. So that was pretty neat. Another really interesting thing here is that while these social parasites have a really solid lock on these bicyclic compounds, particularly the pyrrolizidines, They are not the only megalomyrmex to have these bicyclic compounds. So megalomyrmex myops and megalomyrmex Stud and Jerry also have these bicyclic compounds. Unfortunately, we know next to nothing about their natural history, so it's really hard to hypothesize why there would be this overlap. Sorry, megalomyrmex drifty also has pyrrolizidines. But it seems to be, at least when we consider the social parasites, they're really invested in these novel compounds. So perhaps the bicyclic compounds offer some sort of advantage, or there's a greater piece of natural history that can explain why these compounds would evolve, especially across the genus. For the rest of my work, I use the outline species here for ecological function and physiology: Megalomyrmex modestus, Megalomyrmex petersii, Megalomyrmex mondiboroides, and Megalomyrmex semitochus. I was very unlucky in finding viable colonies for things, so I am condemned to using four/lucky to have been able to find four colonies of, well, four species of what are very rare ants. So getting into the ecological function, one of the most outstanding things, or maybe obvious things for trying to figure out what megalomyrmex use their venom for, is killing insects. I mean, they all interact with insects, so maybe their venom is, well, deadly to them. So to do this, I tested their raw venom. This is, you just tickle the ant and she produces a droplet of venom. You drop it on I used a termite. They're non-co-evolved termite models. They're non-co-evolved with megalomyrmex. However, they have evolved in the presence of other alkaloid-wielding ants, such as Solenopsis. And contrary to maybe what the title of my talk was, we saw that these free-living Megalomyrmex modestus and Megalomyrmex petersii kind of separated on opposite sides of the lethality spectrum. So modestus not very lethal, petersii pretty lethal, while mondiboroides and semitochus stayed a little closer together. And over time, mondiboroides was as lethal as modestus, and semitochus started to get as lethal as petersii, although petersii is still fairly lethal. And I know what you're thinking, Kyle. You said the title of your talk was that the social parasites have more toxic venom. And I think that is still true. And I think that is true because if you separate every conflicting variable and only test the alkaloids. So for this, we synthesized alkaloids and applied them to the termites instead. We find that the parasite alkaloids, those bicyclic compounds, are more toxic. And what happens is toxicity increases as the number of nitrogen bonds increases. So the number of nitrogen to carbon bonds, and toxicity decreases, though not statistically significantly, when using this other metric called molecular complexity. I don't want to get too much into the chemistry here, because that would be like a 30-minute talk all by itself. But you can think of this as a summary stat for things like, if there's the presence of a double bond, the molecule is considered more complex. If the ring is larger, so going from a 5 to a 6-membered ring, that is more complex. These alkaloids have little arms that wiggle off of the sides. If you increase the number of bonds in the arms, the alkaloid is considered more complex. And so what we found is when testing just the alkaloids, the number of nitrogen bonds is significant, going from that 2, or the monocyclic to bicyclic compounds, is significant for lethality, while other metrics are not. I think this is really cool. But what I was most excited about talking about today is this sort of physiology, so extended phenotype. Ants hold all of their venom in a little sac. They have a little reservoir connected to their venom glands. And I took my 4 species, I dissected out the venom glands, or sacs, and that's just really kind of snatch it out, and you try not to feel too bad for killing something. And what I found is, when just considering absolute size, Megalomyrmex semitochus, an obligate guest ant, has a statistically significantly larger venom reservoir, venom sac, then then the free living, the Modestus and Peters Act. Well, that's really neat, especially when you consider down here at the bottom, relative to the other ants, Megalomyrmex symmatocus is not big. It is not a big ant, despite the genus name. Back Symmatocus, I wish I had gotten like a picture of a Peter's eye next to a Symmatocus, but unfortunately they're all dead. Peter's eye is absolutely massive compared to Symmatocus, and yet its venom sac is just larger. But when you control for body size, then these social parasites just absolutely have larger venom reservoirs. And down here, I've changed the size of the gaster, try to be relative to the body size. You can see Symmatocus, absolutely massive gaster compared to something like Peter's eye or Modestus, where they're relatively small. While I was doing these dissections, I just had to take a picture. This is Megalomyrmex symmatocus. You can see on the left is kind of an ant. I've cropped out her head because I was so excited about getting this absolutely pristine dissection done. And you can see here is the stinger, that little kind of lance sticking out. I have a little bit of these last two segments of the exoskeleton. It's pretty hard to take it off without rupturing the venom sac. And then the rest of this bulb is the venom reservoir, their venom sac. And you could think that this was somehow crammed into this gaster. So most of the space in their gaster, which is full of the rest of the organs that they need to live, like their stomach, is committed to venom. And I thought, wow, that's absolutely wild. Their venom is so important to them that they've sacrificed like stomach space because they need to be able to use their venom in order to continue to live with their host. Things like Megalomyrmex Peter's eye need to have that stomach space because they will go out and forage, gather up the food, and then store it in their stomach or in their social stomach. You think of it like a crop from birds, and bring that back to the nest. So they can't have most of the space in their gaster taken up by a venom sac because they need to be able to carry food. To kind of quickly summarize, so in venom alkaloid composition, social parasites have bicyclic compounds, but it's not exclusive to the social parasites. Maybe there's some sort of other evolutionary or ecological explanation. We're going to be trying to investigate that, although I think that might fall a little bit more on my lab mates. For ecological function, the synthetic parasite alkaloids are more toxic, but the raw seem to split. Perhaps this is indicative of something like some sort of specialization, some sort of other metric like antimicrobial activity that we haven't properly tested with all of the social parasites or all of the ants in general, or some other primary metabolite, some sort of protein present. These are being handled by my lab mates. So one of my lab mates is investigating antimicrobial and antifungal activity, and we're getting a visiting scholar in a week, I think, who is going to be looking at primary metabolite or protein presence in these venoms. That's going to be really exciting. And for the extended phenotype for the physiology, social parasite venom reservoirs are much larger compared to free living, especially when you consider ratio, so the venom reservoir to body size. So thank you for coming to my talk. I'd like to acknowledge all of these blurry people in the pictures, my lab mates, my advisor, of course, my various forms of funding, the Smithsonian Institution, particularly STRI, and my fencing club for being an outlet to all of my pent-up PhD candidate rage. Make sure to send hate mail to my email down here.
Thank you very much, Kai. Really, really interesting talk, and to see so much different usages of venom is always super impressive. I had a few questions, but then your last slide came and then you said a colleague is working on this and a colleague is working on that. And so maybe all these questions will be, I think, answered in soon papers. So I will want to have at least one of the questions asked.
Yeah.
You said about the venom to volume range of the ants. Did you calculate them already? Because it looks like it's, I don't know, 25% of the whole volume would be just venom for this one picture you showed there.
Oh, that is a good question. I did not calculate the sort of venom sac to rest of the gaster ratio. This was a venom sac to kind of overall body size. Ants are weird in that their morphology can be incredibly variable. So there's a metric called Weber's length, which is kind of used as the general metric for ant body size. Although megalomyrmex, they're all generally shaped the same. So maybe gaster would be, or gaster length would be a good metric. See, Leonard has his hand raised.
Yeah, Leonard?
Yes, thank you. I think maybe I overheard it, but you said that the parasitic ant is inducing submission by pain in the host ant. But is there also maybe a change in behavior of the host ant towards parasitic ant or something like this, induced by the venom?
That's pretty unclear. When parasites first infiltrate, so a young parasite queen will try to find a young host colony, usually less than 5 workers, because if there are too many workers, they just rip her apart. And the first thing that they do is just try to hide. So they don't want any sort of altercations or anything like that. When it comes to blows, and it very often does, uh, parasites deliver their venom, and the immediate reaction is that hosts will kind of curl up and start to submit. But it's this ongoing, almost courtship in a way of like fighting one another, um, that hasn't really been well investigated. We have some artificial introductions that seem to show that over time, fighting decreases and hosts kind of start to tolerate the parasite. But in our experience with lab colonies, any sort of stress, which could be as simple as like jostling the colonies the wrong way, leads to fighting. And so the behavior is pretty unclear, especially to me. I don't study behavior all too much.
Thank you. And another question, which is a little bit more extended in the ecology, but I learned at some point that some other ant parasites use pheromones to hide within colonies and try to sort of fit in. Did you also, yeah, detect any, or is there anything known about that with parasitic ants?
Yeah, so ants use these, I'm now forgetting what they're called, but they're compounds that exist on their exoskeleton. They're sort of these waxy, cuticular hydrocarbons is what they're called, CHCs. And the parasites seem to have a more muted profile. There's no like notable smells or peaks, at least when we're looking at it. Although hosts are really able to detect parasites when they're interacting with them. So I don't think there's any sort of chemical hiding or insignificance here, at least in these systems. And parasites, even though their profiles are muted, I don't think that the parasite cuticular hydrocarbon profiles and the host profiles overlap a whole lot. So I don't think there's a lot of mimicry going on. So in my opinion, it's kind of about their venom. They're using their venom to kind of create this standoff situation.
Thank you very much.
I have one question about the physiology of the venom system or the venom sac gland. Was it anyway investigated how different the cell wall or the surrounding tissue is shaped? Because I mean, if you have a proteinaceous venom, then you need some normal kind of cell barrier that you don't intoxicate yourself with a protein. But having something that is alkaloid resistance needs some totally different chemical barriers because they are so small and can way quicker interact with the physiology of an insect. So is there anything known about differences between these?
No, that's a really great question, and I can unfortunately tell you that no, nobody has investigated these in any sort of capacity. I think you know, snake venoms are really big. I hope that ant venoms can get to kind of that situation where everyone's just asking and figuring out every single thing about them.
Because I think with the physiology, it's super interesting. They develop these different kind of sacs and the size of the sac. Really, really
Very.
interesting to see so
I
much
think
about the ants.
the kind of only thing that I know about is comparisons between the actual venom gland length. So they have these two filamentous glands that attach to the sac, and the alkaloid ants, so like anything in Solenopsidini, Solenopsis, Monomorium, Megalomyrmex, they have these very long filamentous glands, and in other ants, those glands are significantly shorter. So one of the ideas is that making this alkaloid is sort of a very long pathway, and the starts at the distal end and gets modified and modified until it gets to the reservoir. I think that's the only thing anyone has bothered to investigate or even compared.
So the metabolism and how it's formed is also not known. What is the
Uh,
origin of the? That's
yeah,
super
not at all.
interesting.
My one of my committee members really wanted me to start investigating that. He threw a bunch of chemistry words at me that I didn't quite understand. But maybe someday.